continue the introduction, and again the videos will be online, as you know, some of you have seen. I'm sorry that we have not had all the talks online. I made a mistake in sending the wrong talk, so that's why today's talk is not fully online. Last week you heard about amino acids, protein structure, bonds and energies, and today another constituent of proteins, domains. And next week we are going to talk about the comparison of three-dimensional objects. Protein domains. But let's first recap on what you learned last week. The story, the dogma of molecular biology, from DNA to RNA to proteins, and to protein function and protein structure. Again, DNA is the information, the double helix. RNA is an intermediate step. This action here is shown here. Protein is the machinery of life. DNA is made up of 4 nucleotides. RNA is made up of 4 nucleotides, so that's a 1-to-1. And then proteins are made up of 20 or 21 different amino acids. The first step, biologists like different words. So the first step here, translating DNA to RNA, is called transcription. And the second step, translation. Let's just look at transcription in a movie here from the Virtual Cell Animation collection. So again, this is the step going from the genes, from the DNA to the RNA.
DNA, transcription factors, RNA polymerase, and ATP. Transcription begins with a strand of DNA. It is divided into several important regions. The largest of these is the transcription unit. This portion of the DNA will be used to produce RNA. Upstream of the transcription unit is the Tata box. An enhancer region may also be involved. Several complexes, known as transcription factors, are required for successful transcription. The first is TF2B, the largest of the general factors. A component of this factor, TBP, binds to the DNA using the Tata box to position TF2B near the transcription initiation site. Other transcription factors, including TF2A and T.
Oh, sorry. Somehow I can't get the control. Ay, merde. Sorry for that. I can't control the video. What I wanted to show you, let me just jump in and comment a little bit. So the first important comment here, this is not about you learning all these words that you see. Biology is a lot about words. It's just visualizing a process. Again, we are going from DNA to RNA, and there are different parts that you need for that.
Let's see the process in action.
You begin with the RNA or the DNA.
mRNA, the substrate for translation, has a unique 3' end called the poly A tail.
I made a mistake. I showed you a translation, not transcription.
mRNA also contains codons that will encode for specific amino acids.
So again, codons are 3 nucleotides.
5' end. Translation initiation begins when the small subunit of the ribosome attaches to the cap and moves to the translation.
This thing consists of proteins and RNA.
tRNA is another key molecule. It contains an anticodon that is complementary to the mRNA codon to which it binds. The first mRNA codon is typically AUG. Attached to the end of the tRNA is the corresponding amino acid. Methionine corresponds to the AUG codon.
So you see, this brings the amino acid, and it binds, recognizes the RNA triplet. So 3 nucleotides. In this case, AUG.
The first tRNA occupies the P site. The second tRNA enters the A site and is complementary to the second mRNA codon. The methionine is then transferred to the A site amino acid. The first tRNA exits, the ribosome moves along the mRNA, and the next tRNA enters.
So what you build up here is the protein. Now this will be the fourth amino acid.
These are the basic steps of elongation. As elongation continues, the growing peptide is continually transferred to the A site tRNA, the ribosome moves along the mRNA, and new tRNAs enter. When a stop codon is encountered in the A site, a release factor enters the A site and translation is terminated. When termination is reached, the ribosome dissociates and the newly formed protein is released.
So I'm sorry for gobbling up. So the first one is transcription, DNA to RNA, and I interrupted that movie. The second one is translation, going from the RNA to the protein. Here again, the whole process. Picture of David Gutzel that shows some molecular machinery, so large aggregates of proteins. That's the size of a virus. Those are large machineries, so aggregations of proteins in here. This is not, here's the scale is reduced, as you may see from the letter size here. Those are very long fibers, muscle fibers for instance. Simply aggregates of proteins. Okay? Those are different structures, and structures, these different shapes, they have different functions, and that is what this lecture is about. Let's talk about reality and images. So what I showed you in the last slide was a drawing from David Gutzel. Before an animation. You can't make a photo of that. You can make photos, so to speak, of proteins. That's very complicated. We talk about that in one of the next lectures, how you experimentally determine their structure. It is very expensive and takes a long time. Now, here issues about reality and images. Most of these slides are from Marco Punta, who is now at PFAM. He was a fellow at the IAS. What is that? Wow. It's amazing. Yes, you're totally right. So I don't know whether everybody heard that. So I've never experienced, this is the largest lecture I'm giving, and it's amazing at a size of about 200 people. There's always one in the room who knows something. When you transfer this to a smaller group, if you don't recognize that, don't feel strange. Most of these things, many people in the room will not recognize. So it's the exceptional 1 or 2 or 3. How's this? Where is that? You have seen it before. It's another image from David Gutzel.
It looks like the inside or like a virus?
It's the inside, so very close. In fact, this is the inside of a cell. And one of the important issues, why I'm showing that again, is we imagine cells to be bags of water or something. Most in us, as I told you, two-thirds of me is roughly water. But cells are not bags of water. That's the level of density you have. This is the level of density in a solid state. This is a cell. Could be a virus. In fact, that is the smallest cell, Mycoplasma genitalium. That's a eukaryotic, a cut through the eukaryotic cell. And again, plant cells, animal cells, both have nuclei, have some detailed differences. What is that? Anybody who doesn't know, can you guess? Mouse?
Molecules.
Molecules. Yes, that's true. Do you believe they're connected? What is that? Okay. It's a potassium channel, yes, that got a Nobel Prize, blah, blah, blah. So that's a protein. A membrane protein is at that. And the rods you see are alpha helices. Andrea briefly introduced them. We will get back to this representation of alpha helices again and again in our lecture. It is in fact the same picture. It's just a different representation. While here, for every single amino acid, you have one is represented by one ball with a stick. Now here you see a long rod representing, in this particular case, something in the ballpark of 16 or 17 consecutive residues. So you form structures. If you are used to proteins, then you might most likely see these helices in there. And most people who work on these will in fact recognize this as this particular membrane protein. But again, so yes, you are right, those are molecules, they are connected, and they have a particular shape. As you see, incidentally, in this shape here, it is a little bit more open at the top. Can you sort of guess that? And that in fact is because that's an opening. It's a channel that lets in potassium. And it's opening up here, and the opening is regulated by this little bluish, reddish loops that are next to each other. So they close and open the channel. There's more to it. There's more movement. We will get back to that too. Here another molecule, ADH, alcohol dehydrogenase. And that is in fact the molecule that is involved when you had a little bit too much the day before. It helps you. Is the enzyme that not all in the room have at equal levels, which is in fact the one of the main reasons why different entities react differently to alcohol. Is this enzyme here? What's that? Now anybody can, except for the ones who know. I'm not sure. Do you know? Jonas may know, but anybody? Just throw things at me. Clan. Flam. Flower. Oh, I'm sorry. Oh, I'm sorry. I thought you were hungry. Flower. I like flower. It's not a flower, but it ultimately has something to do that will be misleading. I want more ideas. But for me, flower makes sense. But don't forget about what I said. Just think about something totally different. Yeah?
Fire.
Anything else? Fire, flower, anything else with an F for the game's sake here? So it's not a flan. For those of you who don't know, flan is a nice dish, dessert, caramel pudding or whatever you call it.
Fiber or tissue.
A fiber or tissue. That's because we're in biology? Or do you really see that? Do you really see a fiber?
Yes, it could be like a cloth.
Oh, I get your point. But that is actually, you're sitting next to each other, all three of you. So like these flowers that you make of cloth. I've seen that recently in some museum. I can't remember where. Umberto Boccioni, dynamism of a soccer player. So the fire clearly had to do something, flower activity, and in fact, let me give you a little bit more of a hint. That's one representation of reality. Here's another representation of reality. In fact, that is the photograph that has been used to render this slight derivative of the original image. What I find remarkable, in any sized room, you find people who somehow, the flower, it sort of grows at you. The fire, there's energy. And it's not really clear. I mean, fire, there's a lot of blue in there. Why do you guess fire? But many people come to this idea of something dynamic. That's remarkable. So they clearly, the artist captured a spirit, although arguably those are not really one-to-one matches, those two images. But there's something captured. And it's a little bit the same thing in the world of proteins. So the way we look at these are, in fact, different images. What I should have had here is the molecular view again. I was too lazy. This is what we saw before, the level where I showed you the rods. In this particular case now, they are not rods, they are arrows. And these arrows there are two types of secondary structure. The rods are the alpha helices. The arrows here in blue, in this bluish color turquoise, are beta strands. And the point of the arrow, so F here points in that direction, is the directionality of the chain. Here we have a little bit more detail. We don't have the guiding arrow, but now between these two representations, I guess most of you in the room will see that these are images one of the other. Here's an additional reality. You see there are many different lines. Anybody any idea why there are different lines? Now, for that answer, you need to really know something already about biology.
Are those several captures superimposed over each other?
Yes, essentially, this in fact is different models, if you want, of the same protein. Now, the word model here does not mean that somebody takes a sequence, runs a computer, and gets out that model. That word model here means you in fact have done experiments for 1 year. And your experiments are compatible with two different, or in this particular case, I don't know how many, but this is certainly in the order of 10 different traces, 10 different lines. And they are models in the sense that all the experimental information they have been gathering for 1 year is compatible with all of those. So that's the lack of clarity. That's the degree of error you have in that. And again, you see that you could imagine this arrow here, this sheet, to approximate in some sense this. What is not approximated here is that in this region you see there's the largest deviation, or here's a large deviation, while this is a single loop. And it's in fact not easy to show that on a picture without losing the clarity. Again, I'm going to show you many pictures on many different levels. Be aware, this now in terms of the soccer player is one reality. All of those you have seen a soccer game or have played it know that the 2D image is also not enough. I mean, you guys are familiar with social media. You must have had the experience that you had a photo of somebody, you meet that person, and that person in front of you in 3D looks very different. That's the match from 2D to 3D. There are so many different realities. And so even this one is not the true representation of that soccer player. If you saw that player in action, you would say that this shows nothing of the activity of this guy. And that is the same in many ways in the world of proteins. It's just one view, one reality that helps us think about it. Like the drawings from David Goodsell. The cell is dense, and that's important to think about this. Now, back to the issue. Components of protein structures are protein domains. The molecular components, as I said, are the amino acids. And you know that proteins differ very much in length. But there are other components, and they are called domains. Now, let's get back again to the gene product, or the gene itself. Up here is the gene, and the red regions here are exons. Again, don't worry about those words. Good that you know now that what a gene is, hopefully, after this lecture. But there are regions in the DNA that are actually not coding for a protein. Coding means that are actually not used to be transcribed and translated to a protein. They essentially contain information that in some sense is thrown away. It will not end up directly in the machinery. Only the red ones will be. These red ones will form the gene product. One very early suspicion that people had was that one exon, so one continuous region in the gene that will be translated, or first transcribed and then translated to a protein, corresponds to a protein domain. So the idea was that evolution essentially has already on the DNA imprinted the module of a domain. That is not quite true, and essentially it's not true at all. Biology is full of images and approximations, sometimes called models, you could call it words, whatever you call it, it helps you to understand. Most often it's also misleading, still helps you to understand. Now here is something that people continue to believe in. That is that we have situations where you have one protein in one organism, another protein in another organism, and they are merged in some other protein, in some other organism. Merged meaning on the genome, those different regions come together. Could you imagine any reason why that would be an advantage? Yeah?
For catalysis?
You have to speak up a little bit.
For catalysis?
For catalysis, yes, but you can cut it better. So I'm unsure what you mean. So catalysis means cut, right?
To burn this energy protein. In other environments to burn this protein.
Yes, I believe you're on the right track. This is one of those situations when I'm in a room and I test you. I can hear that you said the right thing, or I can hear that you said the wrong thing. I'm a nice guy, so you said the right thing. In fact, they interact. So imagine that proteins A and B interact. And in this organism where they are separated, they have to first find each other. In this organism, where they are along the chain of the gene and are transcribed, translated together, they don't have to find each other. So that may be an advantage. And in fact, that happens. We have cases like that. Let me show you the idea of domain. And again, you see the rods here, and you see these strands, these sheets, these arrows. And you already see, or you may see, that you somehow could imagine that this protein here has two different regions. You see that even stronger. Well, I admit this is not the best for you to see, but if you turned it around a little bit, then you would recognize that there is some similarity here. That, in fact, gives you the idea that you have some element here that is used again in some other protein. It's not quite the AB story that we had before, but it is a similar theme. Let's call it theme. So you have a unit, a motif, something that you see again because it does some particular function. Here, it comes along with something else than in this context. In fact, if you looked at it a little bit more closely, you would see that there are some similarities even for this region here. But then still, this is an add-on. So clearly, even if you called that one domain protein here, and you would argue that this is in fact repeated here, then this still would be another two-domain protein. And you already see in this problem that somehow where you make the cut, is this a two-domain protein? And correspondingly here, this could be a three-domain protein. Is this a one-domain protein? And this a two-domain protein where I cut here? In some sense, it's arbitrary. Do you have any idea where how you could get this arbitrariness out? Look at that, how to define a domain. Look at it. Does anything jump at you? Yeah?
Well, there are two strongly folded regions.
So something is compact.
On the left, there's a region that's folded together, and on the right, there's a...
Do you mean this?
Yeah.
And this? Yes. So what you said is you strongly folded. In some sense, I believe what this means is sort of compactness. Something where you can look and you immediately see there is a difference here. There's a cut in between. There's an open cleft. There's space, right? And that's your immediate definition. You just look for density, for units, regions that are more densely populated than others. In some lecture there was a biologist in the room. Do you have is that person still around to have another way of looking at it, another domain definition that jumps at you from this? Person's not around. Any other idea? So density is one way in which domains are defined. The problem for that definition is we need a three-dimensional structure. Anything else that you can come up with here? That you can guess, yeah? Can you speak up a little bit?
Perhaps if you're looking at it on a linear way, you can count the different types of elements. For example, there's one cylindric element there and four other arrow-like ones.
So let me repeat that, simply because so my belly is bigger so I can speak louder. She said, I believe, that you simply line up these elements strands, helices, you count them up and you compare them. And you see that left or right, you see a repetition of these basic elements. For instance, secondary structure, right? Yes, that's another way of doing it. It's a very common way, and that already goes into the other idea here. Still, in some sense, it's a geometric way. This particular geometric way, you don't really need to see the 3D structure. You can do it from sequence, and I will show you some examples for that. But the other idea, implicitly, I sort of alluded to it, calling this one protein, this another protein, you look for repetitions in other proteins. Units that are repeated. It's the same idea that you phrased, in fact, looking for the linear elements, you implicitly, directly mean repetition, true. But more generally, then you could argue, well, I call this here a domain because it's repeated in many other proteins. And that, in fact, is another way of defining proteins. It's a very common way to define proteins. Here is another example of that, where, in fact, that is much closer to what you said, the linear arrangement of the domains. This comes from Christine Orengo at the CATH database. I dropped the reference here. I'm sorry, Christine. So again, this is a motif. The motif is seen again here. And here you see a bunch of proteins that are exactly aligned as a string. And these elements are counted, and it's seen that some of them are linearly similar. In this particular case, that each triangle stands for a particular region and domain. Okay, so if we don't have a structure, and if all of these assumptions are true, if domains evolve in evolution, then in principle what we should see are situations like that. Again, next week we are going to talk about 3D comparisons. After that we go into alignments. Alignments are methods that essentially start with a sequence and look at all the other known sequences and ask how similar is that to all the others. Let's assume you already knew how to do that. Again, 2 weeks from now. And we started with a protein, this protein here. And we found another protein that is aligned to it. So this is the entire protein, the bar here, the black bar. And you find another protein B, another one C, D, E, F. Now in this particular case, you may argue, okay, what I see here is a protein that has 2 domains. This part here somewhere is repeated in B, C, and F. Here is another domain that in fact is in F and E. And here is yet another domain that is in E and F. So you would essentially see a bunch of different domains, and you would drag them out, not even looking at this 1D arrangement that we had before, not looking at compactness, but simply looking at what other place in other proteins do you see that region. Now, obviously, and so I should have used the animation. One way of doing it is these 2 domains here, and these 2 domains in fact correspond to the situation that I showed in 3D before. Can you anticipate a problem that you could run into? It sort of looks okay or nice. Again, we're talking biology. In the world of biology, this is a clear-cut situation. What could be the disaster situation? Well, if that would be the dream and that would be reality, then there would be no domain identifiable from these sequence elements. Unfortunately, so our group has spent the best of 10 years believing in this model, and after a lot of frustration, essentially we began believing that it's more like this model. Clearly, we have cases that absolutely look like that. I've shown you one before. I can show you many. There are thousands of these cases. But there are many, many, many cases that look like that. And the more sequences we know, the more we see that evolution really walks in small steps. And it's unclear why that is the case. Let me get into some common assumptions and facts about proteins. How many proteins are we having in human? Or do you have in cell of you? 25,000. Anybody feels this is too low or too high, or do you know already? Same. Okay. Oh, my animation is not. I'm sorry for that. 20 to 25,000. How long are they? Anybody has an idea what the shortest protein is? How many residues? 18 amino acids. That really, so 18 amino acids, you may remember I showed you this receptor where you had the long rods that are inserted in a membrane. One of those membrane helices there is about 18 amino acids. Typically we would say this is a little bit too short, but 30 something is already papain, so there's a normal protein. 18 is still typically a propeptide, but 18, 20, 30 something is where 40, 50, that's where they start. Anybody an idea what the longest one is? Again, oh, let me go back. The scale we are talking here is about 1/10 of a nanometer for an amino acid, or for a typical protein structure. We go into nanometer dimensions. So again, 40, 50 is above a couple of nanometers. Now, what do you believe the longest is? I throw, yeah?
100.
100. Anybody wants to get higher than 100? I hear some muscles are longer. That's true. Now we have to distinguish two different things. So the main theme today is domains. And muscle fibers are built up of proteins, but it's not one single protein. They are built up of many, 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 many domains. So hair, same story. So those are fibers that are aggregates of repeated units. And that in fact is another reason why you have domains, because you can do this, you can stack up like Lego boxes again and again and again, the same principle, and you can elongate yourself by that. I'm asking a single protein, a single polypeptide chain that is co-expressed, that is on the same region somehow in the genome. That's a simplification, but let's not worry about that. So the number on the table is, or in the room, is 100. Anybody goes for more? 100 for the first, 100, you can buy it for 100. So there's titin. Titin in mammals is 30,000. Okay? We start from 30 and other guys are 30,000. Okay, that's an exception. But in human, about 5% of the proteins are longer than 1,500. So 1,500, again, this is whatever you put as the lowest limit, but at least 20, 40-fold bigger than the smallest protein, and there's a lot of those. On the one end, you create large constructs by doing domain repetitions, fibers, hair, other examples, the transcription factor units, where you have in fact an aggregation of different proteins. The ones that you saw in the first movie that I interrupted, these big complexes, are in fact aggregations of proteins, transcription factors. So many different proteins are coming together to perform a particular function, and those are often also referred to as machineries. So aggregates that you could, if you could shine light and you could observe a cell in a microscope, you would see these large aggregates, machines. They act together, okay? And then single proteins that do something else. Again, some of these single proteins are very, very large. Oh God, I'm sorry for the animation. The median is around 400. They consist of units, the domains, we talked about that. Now, here are two statements. Longer proteins are rarer than shorter ones, and prokaryotes shorter than eukaryotes, or prokaryotic proteins are shorter than eukaryotic proteins. Any opinion on the first and second statement? Well, the first statement I implicitly already said that the very long proteins are the exception. Yes, you have 5% or something like that, but still 5% is less than 100, so the first is clear. The second statement, are proteins in prokaryotes shorter than proteins in eukaryotes?
I think it's true because prokaryotes are overall much more simple than eukaryotes.
So the statement is yes, because prokaryotes are overall much more simple than eukaryotes is the statement. Are you the biologist? Ah, great. I set you up. The fact is that most biologists would share your belief. And that's why I like it, because it's wrong. But actually, it's still published in many, many textbooks. So much about believing what textbooks say. It's just not true. It makes a lot of sense. In fact, this is the typical biologist thinking. You have a good explanation. In biology, you need this explanation because otherwise you wouldn't understand the complexity of the facts. Biology is so complex. You need a guidance. You need some intuitive understanding. And it's absolutely true that prokaryotes are simpler than eukaryotes. So it absolutely makes sense what you said. But this particular case is not true. And we actually, again, don't understand why it's not true. Because it makes so much sense. Let's get back into, and I'm sorry to set you up. Give me my joy for a moment. I'm sorry for that animation. Oh, you get it again. Whoo. The problem is when I prepared this slide, I didn't take the animation out, and I didn't see that it's an animation. But it's, you know, it's sort of visually appealing to see boxes being drawn. Oi. Okay, here, what you see, number of residues in a protein, percentage of proteins by kingdom. Eukaryotes in blue, that's a little bit of an old slide. We could have more data, so now it's a little bit choppy. Archaea and prokaryotes. One way of looking at my answer to you is, in fact, prokaryotes are a little bit shorter. You may see a little bit of a movement of green toward the left, but not really. The median, the averages, in fact, are exactly the same. There's some tendency here. So in some tendency, the very, very, very long proteins, the proteins with very, very many domains, those are more in eukaryotes. And that, again, your explanation, we didn't know that it would look like that before we saw it. And your explanation that prokaryotes are simpler, we apply the same thing here. We believe this is because eukaryotes are more complex, but we don't know. Okay, it's just sounds good kind of explanation, and you see that here above in the overall sums. That's the sort of 5%, and I was wrong. I said 5% have more than 1,500, or did I say 1,000? Somewhere around here is the 5% I was talking about. I said 1,050. Anyway, so prokaryotes and eukaryotes are essentially similar. Oh God. Can I speed that up? No. Bear with me. Some proteins are multi-domain. Most are not.
True.
False. True, false. So now let's do some sort of machine or intelligent learning of the mentality of your lecturer. I wouldn't sort of talk about domains if most proteins would be single domain in some sense. In fact, most proteins are multi-domain. And I'll show you a picture once I got through that animation. What this one shows here in green is the fraction of proteins that have a single domain. 60% have a single domain. Okay? And that is what somebody said is true, the statement. Something like 6% have two domains, and so forth. So green clearly is why that statement, most proteins are single domain, is true. Blue is entirely sequenced organism. PDB, what that hides that word, is those are proteins of known three-dimensional structure. Blue are just all the proteins we know in some way to sort of infer domain-like regions from those. You see that in the blue. In fact, the statement no longer is true. So that's, somebody said true, that's the person who talked about the green. Somebody said wrong, that's the person who talked about the blue. And looking at the blue, you see that 70%, so 100 minus 30 something here, or 29, 70% are multi-domain proteins. In fact, there are almost as many proteins with 3 domains as there are with 1. The reason why green is so much higher is it's much simpler to determine a 3-dimensional structure for a single domain protein. That's why in today's database of known 3-dimensional structures, they're highly overrepresented. Simply easier to get at. In reality, most proteins have several domains. And oh God, I'm sorry. How can I stop this? Just jump to the... there are more proteins. That actually it was a mistake, but in there most proteins have many domains. And do prokaryotes have more domains than eukaryotes? Again, biologists tend to believe that the answer to that question is no, that prokaryotes have fewer domains than eukaryotes, and that is not true. Eukaryotes have very, very long proteins, so many proteins with very, very, very many proteins, very, very many domains. But in fact, overall, the number of domains, the distribution that I showed you is very similar between prokaryotes and eukaryotes. And that is essentially what this graph says, which shows the number of, let's call it domains, versus protein length. And you see there's almost, for eukaryotes and prokaryotes, here there's almost the same relation, the same slope. Which means what you can see here is that on average the first domain is longer, every other domain is about 100 residues long. So the answer that proteins are typically maximally 100 residues long, that in fact is most domains are about 100 residues long. Okay, so you can imagine proteins somehow, one way of looking at it, in particular example of an enzyme, you have a larger domain of something that is like 400 residues long, and then you put many, many, many other domains on top of it. And one interpretation of why that is the case is those are different regulatory units. So they allow to switch this main region on and off in a different environment. Okay? That's somehow one of the assumptions that we have in domains. Now, there is this space of structure. What I mean here is that islands are proteins that have a similar structure. There's this space of function. What I mean here is that proteins on the same island have a similar function. Sometimes proteins that have a similar structure have a similar function, but not always. So this is similar structure, very different function. And the same is actually true in terms of experimental feasibility or in terms of sequence space. Things that have similar sequences have similar structures and similar function. But there's a lot of derivation from this statement. One important realization: biologists think about proteins as families. Families of proteins are proteins that are related. They are related in evolution. They are related in terms of structure, the shape. They look similar. And they are related in terms of what they do. Okay? Now, what we have to understand is that these families are, again, reality and image, or reality and the way we represent the reality. Putting these circles around means classifying them by similarity in function, similarity in structure, and always this is a judgment call. There are always exceptions to this rule. This is an interpretation. In reality, proteins evolved. And proteins evolved in a way that you could argue, okay, these proteins in this one here, they are in fact related. That is the same original gene that started 200 million years ago had some basic function that was important to bind RNA, because we evolved from the RNA world. Proteins evolved as slaves in some sense, most likely to the RNA. Most likely that's another way of interpreting why we have 20 amino acids, why domains look the way they look, why the proteins act the way they act today. And so maybe those are in fact those ancient, is one of those ancient regions. And you can clearly trace the evolutionary history of that particular protein. So by that force you can say, okay, they are evolutionarily related, so that's a family. The trouble is, and again, I showed you that before when I said dream of reality in protein domain evolution. The problem is that you sometimes can, somebody said the minimal protein is 18 residues long. You can add a single helix. And with that single helix you can knock out function sometimes, or change function in a dramatically important way. And you can, in a simplified way, you add these 18 residues somewhere at the termini, at the beginning or the end. But very often, in fact, you insert it somewhere in between. Very often you insert it right in the middle of some domain and change by this the domain. And off goes this idea that you can recognize evolutionary relation, that you can recognize structural similarity. So this grouping again is a lot of biology. There are overlapping circles, and the circles have a meaning. The word family has a meaning only in context of a certain question that you ask. Okay? You have to always define what you want. No. What is that? There's something missing. How many proteins are known? 20. We have 20,000, 20 proteins are known. Did you mean 20 or 20,000? So, never again, I'm sorry. And I'm the one who tells everybody in my group, never use animations. Oh. Okay, what I show here is the year versus number of sequences in a database for different databases. PDB, I already talked about, that is a logarithmic scale. PDB, the proteins of known three-dimensional structure. SwissProt, red here, is the database for which we know the function or something about that function. There's somebody has done an experiment on that particular protein and has documented in a paper something about that protein. That's red. Blue is proteins for which we know only the sequence. So clearly it's not 20, so we are much higher here, and I'll get back to that in a minute. I want to point out to you that this starts in 82. This is when the first molecular databases started, and it ends over 10 years ago. Actually, it's 2004. Oh, March 2004. It's 10 years ago that I did that. And you see that already then you had an exponential growth. Plus, what I show here, overlay here, is the doubling every 18 months of the computer speed, or the computer hardware, or the computer ability. That then in 98 was slightly changed to slow down a little bit. That's why this line has a little bit of a dip here. But what you can see is that at some point around 95, the blue is outpacing this light gray. So until what happened in 95 is that for the first time we sequenced entire organisms. The first one was Haemophilus influenza. Yeast was the first eukaryote that was sequenced a year later. So that was the onset of the large-scale sequencing era. And you see that is when, in fact, biodata begins to outpace the computer speed. For those people working in computational biology, that in fact is the nightmare of the story. More data is great because we need more computers and we have more reasons to exist. But we're actually fighting a losing battle because in fact, every thing you do in computational biology starts with a sequence comparison. You take one sequence and compare it against the database. And you immediately see that not only do you lose the battle, but you lose it, you square the loss in some sense. Because comparing one against everything is squaring the problem. So the simple number already outgrow computers, and now you have to square this outgrow. That is the challenge in the field. Here again, the computer growth. In terms of a linear scale, today UniProt is the database of all protein sequences for which we have some idea that they exist that way. There are over 52 million. You see that they double every 18 months now, which computers do no longer do. So the last double here. And in fact, the speed at which they double increases. So computers slow down. You see this is 18 months, this is 20 months here. It gets faster and faster and faster, while computers go the other direction. I'm sorry. The last question is, do prokaryotes and eukaryotes actually use the same amino acids? I've already made this point that they're in eukaryotes there are 21. That's an exceptional case. That is cysteine. That's not really the thing I want to talk about. If I plot overall the amino acid composition, is that the same for the simple prokaryotes and for the more complicated eukaryotes? What do you believe? Same?
Yes.
Why?
Because those are the basic building blocks.
So different elements, metal or any molecule that you see in this room has different use of different atoms.
Well, no, it's all carbon-based.
Yeah, no, sorry. You got me. No, when you look around in the room, is what I mean. You have very different materials. They are all made of atoms. Same principles. But they all have a different composition of the atoms they are made of. The wood, the metal, the cloth. All of these have a different composition if you boil them down to the constituents. Now you argue in the world of organisms it should be the same. I'm only trying to kill your argument. Science is about coming up with an explanation and then trying to shoot it down yourself. If you don't do it, reviewers will do it for you, or your supervisor or your colleague. Find as many people as you can who can shoot it down. The more your argument survives, the stronger it is. That doesn't mean it's always better, yeah?
I have another argument.
Yeah, okay.
That there is a direct relationship between DNA and RNA.
Yeah, but the DNA, so the statement is the amino acid will be similar because there's a clear one-to-one from DNA to amino acids, and since we all live off the same genes, that's the argument, right? Yeah. But it could be a different DNA.
The codons are the same.
Are they? Codon bias? You're the biologist.
No, but you have certain codons.
Yes, but actually even those are different. That is, but you cannot know that. So that is unpublished. But even codons differ. But still, I try to shoot you down. The fact is, the amino acids are essentially similar between archaea, prokaryotes, and eukaryotes. So again, the height of the letter is the percentage of that amino acid. It's the 20 amino acids here. Leaving out the cysteine. And you see that essentially, overall, over the entire organism, they are similar. I'm not going to go through the animation again, but if I went through the animation, then I would come to there's a paper. You can actually machine learn from the composition of an organism to predict the organism. Okay, there is not a lot of functionality to that, but it means that in fact, in detail, the organisms differ very much. Differ enough for you to look at the composition of, as long as the sample set is large enough, you can predict the organism from which you got that. Trouble is that the sample must be really, really large. Now, that brings me to another issue. So, in the beginning, we sequenced entire organisms. Metagenomics is sort of the next way of doing it. Does anybody in the room who is not the biologist know what metagenomics is? So the idea simply is, if you go out there and dug into the ground and took out all the living material that is in the ground, or if you put up something in the air, a bag, and collected all the living material in the air, or you drilled a hole into the ice, or you dumped it into the ocean, you took a boat and you drove with a boat around the globe, and every day you picked samples and just simply sequence everything you find. That's metagenomics. So the idea that you give large groups of environmental things and you no longer care about the organism. The problem is we cannot really now, in fact, from the material we get, see what's in there. But most of the data that is today created is like that. We just have a bunch of DNA, we have no idea which organism it belongs to. But the idea is that ultimately over the next 10 years or whatever time it will take, we will, with this, learn to understand how things work together. Symbiosis is one of these ideas. The fact is that if you dug into this ground here or took it from the air, most of the stuff we find, we have never seen before. Most of the stuff we see there appears to be indicating that there are organisms out there that have not been described. Don't even have to go to the Amazon. You can do that here. That's amazing. So there's a lot of variety, but essentially the point is that the underlying building blocks in this particular case are in fact similar. Let's get a short insertion how we can predict domains. And it goes back to what you said about the linear, what I mentioned about the evolution. So the idea simply is you begin, what the bar here indicates, this is your protein sequence. Now I said domains are also characterized by some density. Looking at it, you saw where you can cut, remember? So when you have a three-dimensional structure, that's what the word PDB, protein data bank, means, that you know a three-dimensional structure. You can identify domains. Here we have two domains. They are, just looking at the sequence, you find that they are similar. So you say cut, okay? So I would argue this is one domain. This region here is less than 18 residues, so I know it cannot form a protein, I throw it away. Okay, now I have this one here. The next step is oh, you guys are scary. Wow. Today I have it with my animations, huh? My problem now is that I do not at all see the same thing on the screen that you see on the... Here's the next way of doing it. And again, I'm going into 2 lectures from now. In 2 lectures from now, I'm going to talk about how we can take a sequence and compare it to another sequence. Alignment methods. There's a database called PFAM, Protein Families, that has been started by a couple of people. I'll give you more details on PFAM at the later point in the lecture. But one of these people is Alex Bateman. Alex Bateman essentially started in 1995, came to work and thought, let's build families. Families are things that somehow look alike in the way I showed you, overlap, similar alignment in different species. So the way he did it is he used software and he read a lot. And after a week, he had 1 new family. And after 2 weeks, he had 2. And then he got faster and he built up family by family by family by family. And essentially he has been doing that for over 10 years, the group has been growing, so now we have 10,000 of these. Again, every single family is built by somebody who comes in the morning, runs some software, looks at the results, compares it to the literature, reads about it, and at some point decides, now we have a new family. So those are assignments or curated assignments of domain-like fragments. So I run the remainder of my protein here against PFAM. Let's assume that I find a PFAM region like this, I cut again. In this particular case, that one here would be left, that one. So I have one domain, two domain, and a region that is left here and a region that is left here. Let's assume I find something like that in a database of proteins for which I know they exist. I have some experimental information here, Swiss proteins that used to be the red in the graph I showed you. And you see some hit to these proteins, some hit to these proteins. Again, in the spirit that I showed you before, you assume that these are two different domains. And you cut. Oh God. No. So in the spirit of Easter, mea culpa, mea maxima culpa. In this particular case, you would end up with 5 different domains. When you do something like that, then you see that in fact about 1/3 of all the regions cannot be assigned. 2 domains. That is a simplified way to assign domains. The story that I didn't address, let's begin right there. We have 52 million protein sequences known. For how many of those do we know the structure, the 3-dimensional structure? Let's begin with a few numbers here. Anybody has an idea how you in fact determine the structure of a protein? So you cannot look at it with a microscope, okay? There are 2 different techniques, 2 main techniques, NMR, nuclear magnetic resonance, and X-ray crystallography. Let's begin with the one that produces most of the known structures, X-ray crystallography. So you take the protein and you find a way to make this protein grow as a crystal. This is not as surprising as it may sound because, again, the density level in a cell are solid state kind. So proteins grow crystals. So you grow this crystal, then you shoot X-ray beams into it, and you need a very, very good X-ray source for that. CERN is a typical example in Germany, in Hamburg, DESY. So there are a few places in Europe where you can in fact have good X-ray sources for doing something like that. You shoot this from the scatter of the X-ray, you can compute where the original electron density was, where in fact the particles are, the atoms are. And that is a laborious task. Any idea, anybody in the room, what a protein structure costs? Ballpark? Numbers. Money. Time, whatever you want to throw at me. How long does it take to determine the structure? And how much does it cost? So X-ray beam is not something that I can carry around in your pocket. So time, it used to take several years. There are many proteins that have been evading structure determination for more than decades. Particular membrane proteins are very tough because they're very important and very difficult to crystallize. The fastest today, 3 months, 5 months, 6 months is a very, very fast time. But several years still, I believe, would for most new proteins be a good average. Money. Any idea? That's 100,000 is a very good number. I don't know, did you know? So 100,000 is very close. It's a 10 times off, but in principle very close, so it's $1 million. No, you're laughing, but this, you know, you have no idea what it costs. So 100,000 is close enough. And in fact, 100,000 is one of those things that we hope that we get to over the next 5 to 10 years. $1 million is the number that has been reached through large-scale structure determination at the NIH in the US. Over the last 15 years, it came down to the level, on average, less than $1 million. But that sort of is the contemporary number. Today, we know about 100,000 of these. And you do the math, and you get some idea about how much value is in this database, PDB. 10 by the 5 by the 10 by the 9. Now, again, we have 52 million proteins. For about 100,000, we know the structure. Okay? The situation is not as bad, and that is, again, most of my lecture will be to map these two numbers onto each other. But this here is the attempt to somehow find some regions in between. So again, this works for the smallest number, because you have these blue things here only for 100,000. The PFAM regions, I said, you have for 10,000, and we have 52 million. So this is the attempt to somehow map one universe onto the other. Okay, I'll stop here. On next Tuesday, we will go into a 3D comparison. Next Thursday, we will be skipped because May Day. And the Tuesday thereafter is skipped because of the student assembly. And then we will get into alignment techniques, so ways to compare strings of sequence in order to compare protein sequences. Are there any questions about today, about the future, about the past?